Hello everybody, my name is Aleksandra Stevovic and today I'm going to present to you the poster with the title Molecular Characterization and Treatment in Patients with Rare Cancers from our EORTC-Arcagen retrospective feasibility cohort. Rare cancers are cancers with incidence of less than 6 out of 100,000 cases per year. They include more than 300 histological subtypes and they can affect all organs. The incidence in adults is 20%, whereas mortality rate is 30%. Molecular characteristics and treatment options are not really well defined, mainly because they are underrepresented in research programs. Therefore, the goals of Arcagen projects are to understand the molecular landscape of rare cancers, including genomics alterations, tumor mutational burden status, and microsatellite instability. Also, another goal is to evaluate clinical actionability and treatment options for rare cancers. In this cohort, we successfully analyzed 85 samples from 77 patients. And as you can see from this table, we had 41 patients with sarcoma, 9 with ovarian yolk sac tumor, 14 with rare head and neck, and 13 with timomas. Age at the diagnosis was the lowest for ovarian yolk sac tumor, and gender was generally balanced between male and female except for ovarian yolk sac tumor. We had 2 histologies for sarcomas and 5 different histologies for rare head and neck tumors. Most of the samples were from primary tumors except for 1 recurrence in sarcoma, 3 recurrences in timoma, and for 6 patients with timomas we had both primary and recurrent samples. So these samples are analyzed using targeted next-generation sequencing analysis by Foundation Medicine, and there we used 2 assays, Foundation 1 heme assay for sarcoma patients. There we had the DNA and RNA analysis on a subset of genes, and we used Foundation 1 CDX assay for other patients. There we had only DNA analysis on a subset of genes. Molecular reports are provided by FMI and the analysis was done by EORTC. So here on this graph you can see the alteration per gene and tumor type, and as you can see for sarcomas in orange and rare head and necks in blue, most mutations are found in TP53 and RB1 genes, whereas for thymus we have some mutations in HRAS and BRAF genes. And from this graph you can also see that ovarian is very silent, so we couldn't detect many mutations there. Regarding actionability, we could recommend the treatment to patients approved in EU in the patient's tumor type only for 3 sarcoma patients and 1 rare head and neck patients, shown in green. In yellow, you can see the patients for which we could recommend the treatment approved in EU but in other tumor types. And only for 3 patients we could recommend only clinical trial. There are many patients shown in gray for which we could not recommend any treatment. And if we look at the alterations based on which we could recommend the treatment, if it was a treatment in the same tumor type, we could recommend it based on 2 fusions, 1 amplification and indel, whereas in other tumor types and clinical trials could be targeting SNVs, rearrangements, and high tumor mutational burden status as well. And if we compare these results to the results from FMI cohort with common tumors, here shown on the right, we can clearly see that for more than 50% of cases in our Arcagen cohort with rare tumors, we could not recommend any treatment, whereas this number was only 9% in FMI cohort with common tumors. Also, for only 4.7% of cases, we could recommend a treatment approved in the EU in the patient's tumor type in our cohort, whereas in common tumors, this number is more than 50%. These big differences clearly show that there is a gap in the research between rare and tumor types, and maybe that the gene panel analysis is not being adapted to rare cancers, and that the drug and clinical research is not focusing on the rare cancers as much as the common cancers. And so it's more than evident that we need more research in rare cancers, This pilot phase was very successful and recruitment for Arcagen prospective cohort is ongoing, and we hope that with the bigger cohort of rare cancers we could confirm the findings from this pilot phase, and that we can even go further with the research. With that, I want to thank you for your attention. If you have any questions, please leave them in the comment section.